In this video, we'll talk about basic common carbohydrates. To start, we'll talk about monosaccharides. We will focus on our hexoses or 6 carbon rings in this video. We'll focus on pentose when we talk about DNA and RNA synthesis in another video. The most common monosaccharides that we hear about is glucose. However, if we take glucose and form some isomers, epimers, or enantiomers of glucose, we can get our mannose, fructose, galactose, and other monosaccharides. We can also make disaccharides. Disaccharides are when we combine 2 monosaccharides together. For example, we have lactose, which is commonly found in dairy products. This is formed by combining glucose and galactose together. Similarly, we can form other disaccharides in the same manner. For example, maltose is glucose and glucose combined, and sucrose is glucose and fructose combined. These disaccharides are joined together by a glycosidic bond. We have our oligosaccharides. These are ones that are 3 to 10 monosaccharides combined together. The common categories for oligosaccharides are glycoproteins and glycolipids. Glycoproteins is when we have a protein attached to the sugar, or if we have glycolipids, we have a sugar attached to a lipid. Glycoproteins are commonly used as immune recognition. For glycolipids, it is in the name a sugar and a lipid combined together, and we mainly see those in the cell membrane for membrane stability. Lastly, we have polysaccharides. Polysaccharides are when we have 10 or more monosaccharides together. They are linked by a glycosidic linkage, and they can be joined in various ways. So they can join at the alpha-1,4, alpha-1,6, beta-1,4, or beta-1,6 linkage between the different monosaccharides. The common monosaccharides that we talk about are cellulose, starch, which are 2 types of starch, amylose and amylopectin, and glycogen. It is important to know the different glycosidic linkages and which one of these polysaccharides has what to understand its digestion. For example, alpha-1,4 will give you a linear polysaccharide, alpha-1,6 will give you a branched polysaccharide. Beta-1,4 is actually present in cellulose, and it is hard to break down, and actually it is not broken down by our bodies at all. We need to understand how polysaccharides are given to us as humans. So its source, what bonds each one have, and whether they're branched or not. Branching allows for fast degradation or breakdown of the polysaccharide. So if we eat things that are branched, they break down faster, we're able to get that glucose or energy a lot faster. So let's break this down. Let's start with the source. So for cellulose and starch, those are going to come from plants, and for glycogen, that's going to come from animals. For the bonds, cellulose has the beta-1,4 bond, which we said was impossible for humans to break down naturally. We don't have the enzyme in our bodies to break down the beta-1,4. For starch, again, we have 2 different types. We have amylose and amylopectin. Amylose will have an alpha-1,4, but amylopectin will have an alpha-1,4 and an alpha-1,6. That is because the alpha-1,6 is going to be the branching. So amylopectin will be branched. Anytime we see that alpha-1,6 along with the alpha-1,4, the alpha-1,6 will be the branches off of the alpha-1,4 chain. So for glycogen, we have both alpha-1,4 and alpha-1,6. This is easy to tell the branching because now we have branching in amylopectin and glycogen because of that alpha-1,6 again. And we have no branching for cellulose and starch, specifically the amylose for starch. So why is it important that we know about our carbohydrates? It's very important in understanding our gastrointestinal system with everything that we eat on a daily basis. One important thing we relate to in this lecture is going to be cataracts. Cataracts is when there's a breakdown of proteins and fibers in our eyes, causing slight blindness and progressive blindness if untreated. The breakdown can be due to many reasons, but conditions such as diabetes and galactosemia can be a huge contributing factor. For diabetes, Diabetes causes an increase in plasma glucose, also known as hyperglycemia, and if it is left untreated, we have an enzyme in our body called aldose reductase, which will convert the glucose into an alcohol known as sorbitol. Similarly, in galactosemia, we have an increase in galactose in our blood. That galactose is going to be converted by the same enzyme, aldose reductase, in our body into an alcohol. We'll call that one galactidol. Both galactidol and sorbitol are going to affect our eyes by increasing the osmotic retention, meaning we're keeping a lot of fluid in our eyes. That extra fluid in our eyes is going to put pressure on our eyelids, and eventually that pressure is going to cause degradation of our eyes. And that degradation, again, of those proteins and fibers in our eyes is the cause of cataracts. Thank you so much for watching this Moncheez short on carbohydrates. Hit the like button if you enjoyed this video. Let us know what you think by leaving a comment, and don't forget to hit the subscribe button to get notified when we post new material. See you in the next one.